So thanks a lot, Isolt, for introducing quite a lot of important concepts like the difference between metadata and data and the importance of data management. I'm gonna present the NanoCommons knowledge base, and my background is a bit different maybe from most here. I'm a molecular biologist, but I've been working in bioinformatics for the last 20 years, and I'm actually placed at a company in Munich, Biomax Informatics. We originally come from bioinformatics, but moved a bit more into toxicology as that becomes more and more complex as a field. And our focus really is data and knowledge management, and that's the part here. So NanoCommons has a very open data and knowledge management concept, and I'll just very briefly touch on that because if you consider working in this environment and using the tools, it might be good to have it in the back of your mind, even if you don't need any details. But the important aspect is that as an infrastructure, we try to bring together, on the one hand, data providers of different types, from phys-chem characterization to toxicology to model simulation, descriptor production, and data consumers, whether that's regulators or modelers or whoever. And that's where the NanoCommons knowledge base is somewhere in the middle. And there are different aspects to this bringing together. And one of the aspects is that, well, currently, very often knowledge management means someone, a researcher doing a machine learning approach or a modeler doing model validation, needs access to a couple of different sources, and they set up their pipeline, they do their own integration, and that works fine. It's not very reproducible because the next modeler might use a different pipeline, and it's not very efficient because everyone has to set up their own pipelines. So what we came up with as a concept is we use something called a semantic model, which really is an abstract model of the world that you're working in. Maybe those are nanoparticles, of course, proteins, compounds, and how they are associated. And we use that semantic model to integrate a large number of diverse sources. In this case, for example, NanoPhase, NanoMapper, ACE Nano, and so on, based on application programming interfaces, but also on user interfaces for upload if you have data in an Excel file or an electronic lab notebook. And then we provide both a user interface, but also application programming interfaces to enable the access to this kind of integrated information. And one important aspect is that this is not necessarily data transfer. It can be just an integration based on really technical interfaces, the APIs, and then the data stays in your NanoMapper instance, in your ACE Nano instance, or whatever data source you're using, but you're still able to search across many different data sources because you have this kind of semantic matching. That also means if you are using the NanoCommons knowledge base, you might, for some data sources, be able to dig down directly to the original data, while for others, you might be on the level of the information, what kind of data is there, and then you might be forwarded to an appropriate data analysis agreement or to a more in-depth application that gives you detailed access. So you have different levels here. Okay. So overall, the important aspect is that we are not necessarily copying any data into a NanoCommons knowledge base. We're not adding another data warehouse trying to centralize everything. We're really providing an infrastructure that allows access to existing systems. That doesn't mean we don't, as a part of the infrastructure, offer a data warehouse. So if your data is in Excel and you need some more appropriate storage, then that's something you can consider and you can do. And as Isolt mentioned, the transnational access activities allow you to do that as part of a project. But it's also about connecting sources. So if you are part of a project and you have set up an important source for, let's say, exposure data, which you would like to make visible and available to others, then also please consider NanoCommons knowledge base as a tool to make it broadly visible. So this technical or this workflow is really mapped on this technical infrastructure, which provides us an interoperability layer that allows to make available to others a lot of information available in nanotoxicology. And that's also coming back to the original first slide, how the data generation from particle characterization to complex toxicology and the data analysis and simulation that you're going to do today with the Jaqpot is actually connected, because you have this underlying network for interoperability. Now, just a few examples that are currently available in the NanoCommons knowledge base. NanoCommons knowledge base is really fed by a number of nanotox projects, of course, NanoCommons and NanoSolvit on the technical side. Content-wise, for example, NanoMile and NanoPhase. We currently have information for more than or about 500 particles there, with PhysChem characterizations, with toxicology assessments, mesocosm, environmental measurements from NanoPhase, and the protocols for measurements. And also within NanoCommons and NanoSolvit, now more and more in silico predictions of descriptors, corona predictions, and so on are added. Regarding data access, NanoMile data is already freely available. NanoPhase data is embargoed until September 21, but there is a governance and a data analysis agreement available, so you could actually request access, and there is a process for being able to access that kind of information. There's also a couple of tools in the system. The search tools for metadata, which include ontologies like the nanomap ontology, the nanoparticle ontology, but also the NCBI taxon ontology, for example, to be able to search the model organisms that have been used, or the gene ontology to be able to search the biological functions that have been affected, the pathways. APIs, and one example of API integration, of course, is Jaqpot. Tool integration for expression analysis, for modeling, and abilities to generate ontological descriptors for your data and do data uploads. Those two, I have to say, are currently being revised. So right now they are not there, but they're gonna come back within June. Why is this kind of integrated, structured management of data important? Martin and Isolde already mentioned fairness of data, which means it's findable and it's accessible and reusable. Now, in difference to an archive, which is a great way of storing data in a long time environment, and you can somehow browse and search descriptors. If you do this kind of semantic integration and structure your information, first of all, you can harmonize and map data from different sources. So you really have connections there. You can map it to the metadata, which allows you to not only browse, but infer. So you can really use ontologies to infer through them. You can combine different types of measurements, molecular, phenotypic, environmental, and you can visualize. And just to give an example, for NanoMile and NanoPhase, those informations have been mapped not only to particles and their descriptors, but also to an overall background knowledge of genomes of the organisms that have been used for assessment, of pathways that have been available for those genomes, signal transduction pathways, metabolic pathways, protein-protein interaction, transcription factor networks, and so on. So that makes a much richer environment in terms of using the information and making it available. Another aspect, of course, of the infrastructure is connection to tools, and that's going to be the focus today, the Jaqpot tools. I'm not going to go into it. And then there's a couple of services available, for example, mining the data for different particles, for characterizations, for toxicology. That's now the NanoCommons knowledge base user interface that you see here. Data upload and annotation meaning providing ontological descriptors, ideally descriptors as a minimum for your data. Isolde mentioned the metadata. What have you measured? What's the meaning of the variable you have measured? What are associated protocols ideally? We have an integrated service for expression analysis right now. Currently for RNA-seq, if array data would be an important aspect, that's very easy to expand. That's focus on differential expression, on some visualization of the differential expression. It also can provide differential pathway analysis and gene set enrichment analysis. And we have integrated first models, and those will be coming stepwise in NanoCommons and NanoSolvit. For example, we have a corona prediction model that allows you to, for a nanoparticle, predict how specific proteins would form part of that corona. Basically, you select a nanoparticle, 1 to n proteins, and you run the analysis. If we do have 3D structure of the analysis, it's quite fast. If the 3D structure has to be predicted, it becomes really compute intensive, and we would ask you to do that as part of a TA, because we really need to dedicate resources here. If it's something quick, because there is a PDB structure, then that's easy to do. And the result then is the prediction of the corona information on zeta potentials and contribution. Just a few next steps. What's coming up next? We're currently working on integration of external sources. There's a couple of Inanomatter Ambit-based projects, 7 or 8, that use that system for data management, and we are integrating based on the API. There's also a couple using the ACE Nano system. There is Nested, there is the EU1 who are interested in connecting, and as I mentioned, modeling analysis tools. And the second part really is data integration for those of you that do not have any use any structured system right now, and of course for modeling results. And the best way of approaching that would be a TA, really. There's a lot of different projects behind that. I mentioned the nano projects, but in terms of public projects, there's over 20 public projects using that system. So that's really getting input from a lot of different systems, also from the more systems medicine oriented area. Thanks a lot. And just to show you how to access, if you're on the NanoCommons webpage, then you go to the knowledge portal here, and there's a register here link at the bottom of the page. So the knowledge portal now is open for everyone. You just have to register automatically, then you receive access to it. There's no restrictions apart from the ones mentioned, like nanophase data being embargoed or other data potentially being embargoed. Thank you very much, and very happy to take questions if there are any.